Guy? Guy, please.
Hey. Hello, everyone. Let's wait another minute because I see people are still joining the event. Okay, I'll start. So, hello everyone and welcome to the 8th event of AI in Genomics Meetup. Maybe some of you recall, but we had our last event back in January where we talked about AI and the microbiome. It's been a while and I try to wait for a time where we can make a physical event, but unfortunately it's only getting worse in Israel. So eventually there was a great opportunity to arrange a meetup with the help of Luis and Shlomit from Immune AI. And I decided to go for it, although it's virtual. So again, thanks a lot, Immune AI, for helping in arranging this super professional event. On another topic, I'm glad to update that our community is continuously growing with over 1,500 people. So again, as I do in every meetup, I encourage you to contact me in case you want to present or have some initiatives around the topics we discuss in this meetup. So what do we have today? Today we have two speakers, Rebecca Erbs from Immune AI and Omer Weisberg from Harvard, with some very interesting talks. You will also get to learn from Rebecca about the work they do in Immune AI. I encourage you to ask questions during the talk, and we'll have some time for Q&A. So just before we move to Rebecca's talk, I'll briefly introduce her. Rebecca did her undergraduate studies at the Swiss Federal Institute of Technology in Biology and Chemistry. She then moved to the Weizmann Institute for her master's studies, where she studied the genetic basis of hybrid vigor in yeast with Naama Barkai. After her move to Boston to join the system biology PhD program at Harvard, she joined the lab of Aviv Regev. Her thesis focused on applying single-cell RNA-seq to two compelling biological applications. One, studying the anti-tumor T-cell response longitudinally over tumor progression, and two, to study the choroid plexus across brain, ventricles, and development. After her PhD, she joined Immune AI as a computational biologist. Today, our talk will be about studying this function of tumor-specific CD8 plus T cells longitudinally across tumor development using a single-cell analysis. With that, we'll move to Rebecca.
Okay, I hope everybody can see. Good afternoon, everyone. One second. To make it small. Okay. Good afternoon, everyone. Thank you very much to the organizers for giving me the opportunity to let me speak today. So yeah, I joined Immune AI in August as a computational biologist, and before I present my PhD research, I'm excited to tell you about the work we are doing at Immune AI. So the company was founded in December 2018 by Noam and Luis, that were at MIT at the time, together with Ansu and Danny, who are at Stanford and at the Parker Institute in California. On the scientific advisory board, we have leaders in the field of immunology, single-cell technology, oncology, and machine learning. Our lab is located in New York City, and the computational team is here in Tel Aviv at the Sarona market, so we eat very well. We have about 50 employees across the 2 locations at this point, and the company has raised $20 million so far. So now I'll tell you a little bit about Immuni's vision. Immunotherapies have shown tremendous success and sparked a lot of new excitement in the field of oncology, but the response of patients to immunotherapy is highly variable, and we lack good biomarkers. So at Immuni, we aspire to improve both the short-term and long-term response rate to immunotherapy, identify biomarkers to predict efficacy to treatment, and extend the success to additional cancer types. We do this. To do this, we need to better understand the mechanism of action of these drugs, as well as of response and resistance to these therapies. For that, we need to have signatures that are early indicators of whether an individual is responding, so we can intervene with a different therapeutic strategy in case needed, or to predict which patients will show long-term benefits. At Immuni, we believe that looking at the immune system with unprecedented depth will enable us to solve these problems. Our strategy is to characterize the immune response across a large number of patient samples at the single cell level. We focus primarily on blood because obtaining large numbers of clinical tissue samples is not feasible, and because the blood is a great place to look at, as I'll discuss next. I don't think I need to convince you that we need to be looking at clinical samples, nor that we need to be looking at many patient samples. But why look at the blood? Is it the most accessible way? It is the most accessible way to query the immune system, but is it an informative place to look at? More and more evidence is telling us that the cells in the periphery reflect the biology and pharmacodynamics of the tumor, and this is an amazing finding. Together with the accessibility of blood samples, it allows for unique clinical opportunities. Several recent papers have shown that in patients treated with checkpoint blockade, the number or subset of immune cells and their clonality in blood predicts outcomes. These discoveries also underscore the need for multi-omics data, both phenotypic characterization of cells as well as T and B cell receptor sequencing to look at clonality. There's also increasing evidence that in-depth analysis of the blood of patients treated with immunotherapies enable discoveries into the mechanism by which drugs are working. For example, in a paper from Ansu and Danny, both of which are founders of Immuni, showed that in patients receiving PD-1 blockade, a large percentage of the T-cell clones you find in the tumor after treatment come from the blood, as opposed to having been present in the tumor and expanded after treatment. 2 independent studies by a group at Genentech and UPenn show that there is a clear pharmacodynamic signature of drug action in the blood. Now, when looking at the blood of patients, what are the most powerful technologies currently available to profile the immune system? Taking basically all of the benefits of bulk RNA-seq, flow cytometry, and TCR-PCR sequencing and putting it all together, single-cell technology, we can comprehensively profile the mRNA at unprecedented resolution, which we believe you need to look at to drive important insight. We can also couple this phenotypic characterization with information on each cell's T or B cell receptor, as well as protein markers. This is what we have found most informative in what we do at Immuni. So at Immuni, we create an end-to-end platform that starts in the lab in New York, where we improve sample quality, standardization as much as we can. We then build high-quality sequencing libraries, doing site staining for cell surface markers and TCR-PCR sequencing. We create high-dimensional data for many patients, about 1 terabyte per patient. Analyzing hundreds of thousands of cells requires a sophisticated computational infrastructure of machine learning approaches. This computational infrastructure corrects for batch effects across many samples and then leverages our cell atlas of millions of cells to annotate cells with very high granularity. Computationally, what we do is use all the single-cell data together with the clinical and lab metadata and apply integration algorithms we've developed to reduce batch effect and extract robust biological insight. These approaches are related to published algorithms, but are optimized to handle very large data sets and very heterogeneous data that couldn't be analyzed with published methods. These high-quality data allow us then to map things like treatment arm, response type, treatment time point, and signatures onto the cell annotations and clonality data. We can now ask questions about the phenotype of clones that are expanding, cell types that are underrepresented in a subset of patients, or biomarkers for response. And with that, I'm gonna switch to talking about my PhD research. So I joined the systems biology program at Harvard and joined the Reeves lab, particularly at the Broad Institute for my thesis work. And today I'm gonna share with you one part of my thesis, where we studied the anti-tumor immunity using single-cell RNA-seq with a focus on the computational side of my work. So before I dive in, I'd like to introduce the people involved in this project, and most importantly, Jason Schenkel, a postdoc in the lab of Tyler Jacks at the Koch Institute. He's an excellent immunologist and scientist, and he did all of the experiments and generated all the data I'll be presenting here, with the help, of course, of many others in both Aviv's and Tyler's lab. So first, I'm going to give you a short introduction and motivation why we were interested in studying the T-cell response longitudinally, and also which model system we used. And then I'm going to discuss our results, how dysfunctional CD8 T cells developed over tumor development, and specifically how I used latent Dirichlet allocation analysis to analyze the single cell data. I'm giving a quick summary at the end. So the immune response plays a dual role in cancer development. On the one hand, we know that chronic inflammation can promote tumor development, as tumors can feed off the tissue repair mechanisms. On the other hand, the immune system can be an external tumor suppressor as it detects and destroys transformed cells. This dual and important role of the immune system has been well acknowledged, as you can see here in a revised version of the Hallmarks of Cancer from 2011. However, the idea that the immune system can play a protective role is quite old, and it took a while to perform clean experiments to show that. In the 90s, researchers generated a genetic knockout mouse that was lacking the adaptive immune system. And this mouse model allowed researchers to elegantly show that mice that were immunocompromised developed more carcinogen-induced tumors, as you can see here in red. Furthermore, they showed that tumors that developed in immunocompetent mice were more aggressive. And this insight that immunity can shape not only tumor quantity, but also quality, informed the idea of immunoediting. A proposed model of this process of immunoediting looks like the following. Initially transformed cells are detected by the immune response by their neoantigen expression and destroyed. If a growing cancer persists, it can enter an equilibrium phase that might last quite some time. And during this time, cancer cells are selected for their ability to either evade or suppress the immune response, and can eventually grow out to form fully malignant and pathological tumors. Therapeutic advances in cancer immunotherapy basically try to reverse this process and to establish a functional and vigorous anti-tumor immune response. There are several approaches to boost anti-tumor immune responses to induce durable tumor control. Current approaches include the blockade of inhibitory receptors, such called immune checkpoints, and these have yielded unprecedented clinical benefits in several tumor types. What are these inhibitory receptors? The activity of cytotoxic T cells has to be carefully regulated and tuned in order not to overshoot and cause too much tissue damage or autoimmunity. But on the other hand, they need to clear an infection properly. So co-receptors can either stimulate or inhibit signaling through TCR, a few of which are listed here. An early target of immunotherapy was the inhibitor receptor PD-1. And here you can see a Kaplan-Meier curve for non-small cell lung cancer that shows the improved benefit of PD-1 therapy in blue over standard chemotherapy in green. Clinical responses to immunotherapy correlate with pre-existing CD8 T cell infiltration, the mutational burden of the tumor, and of course, ligand expression. However, there exists tremendous variability, as I mentioned before, in response between patients, and we don't have currently good biomarkers to fully be able to predict response. This highlights the urgency of better understanding the dynamics of an evolving anti-tumor immune response. So at the beginning of my PhD in 2013, I wanted to study anti-tumor immune response longitudinally from early lesions to advanced tumors. However, this required a proper model that allowed us to study this process. And this is when Aviv and I reached out to the JAX lab, who have developed a great mouse model, which is called the KP model. It's a genetically engineered mouse model for lung adenocarcinoma, and it's induced by lentiviral delivery of CRE to the lung epithelium, which induces expression of oncogenic KRAS and the deletion of the tumor suppressor p53, hence called the KP model by its genetics. While transplantable tumor models in mice have been important for our current understanding of T-cell dysfunction, these models have several shortcomings in how they recapitulate critical aspects of human cancer. In particular, they are rapidly growing, lasting only a few weeks before the tumor reaches a size in which the mice have to be euthanized. Second, tumor cells are injected into a non-native tissue site, most often the flank. And third, thousands of millions of cancer cell lines are transplanted, resulting in non-physiological disease progression. These are significant hurdles and have real ramifications for tumor-specific CD8 T cell differentiation. So the benefits of this model are that it develops over several months in its endogenous tissue, allowing for a longer time for the T cell response to mature and evolve in its native environment. However, in order to make this tumor immunogenic, an artificial antigen termed leuco-S is delivered together with CRE. Leuco-S stands for luciferase, fused with chicken development, ova, and another peptide, SIY. So with this antigen in this model, as you can see here in black, tumor growth is delayed, which is a T-cell infiltration dependent mechanism, as this effect is lost in Bragg knockout mice, as you can see in the middle graph. But the number of anti-tumor CD8 T cells declines over time, and tumors nevertheless progress, in spite of continued antigen expression. So what's going on here? We can learn from chronic viral infections where the infection can be cleared and antigen persists. We know that in such settings, CD8 T cells become so-called dysfunctional and eventually lose effector functions in order to protect the host from tissue destruction. During this process, CD8 T cells progressively lose their proliferative potential, their ability to secrete cytokines, and they upregulate inhibitory receptors. So can we see this progression in our model? Indeed, we saw all these classical progressions associated with dysfunction during tumor development. So tumor-specific CD8 T cells peak in their ability to proliferate and secrete cytokine at 5 weeks post-induction, and they progressively upregulate the number and level of inhibitory receptors expressed with time. I'd also like to point out here that we quickly learned that the shift and that the most dramatic shift we saw was very early on during tumor development, between 5 and 8 weeks, when the tumors are still microscopically small. So can we identify transcriptional programs that are associated with these functional shifts, and what can we learn from them? To study the anti-tumor immune response dynamically in depth, we induced the tumors and we characterized the lung-resident tumor-specific CD8 T cells at several time points after using single-cell RNA-seq, flow cytometry, and functional analyze. I'd like to point out that when we isolated the CD8 T cells, we can sort them for their ability to recognize the tumor antigen, and that allows us to study specifically the tumor-specific T cells. While we originally studied the process up to 16 weeks post-induction, I'll present here results when we focused in more detail on the dynamics early on, as we learned that the cells are already dramatically different by 8 weeks. So here you see all the tumor-specific CD8 T cells in a UMAP embedding profiled at 6, 7, 8, and 12 weeks post-induction, and they're colored by the cell density on the UMAP at the respective time points. And I'd like to take here a little detour now, and we'll discuss how I use probabilistic models to analyze what I call a more continuous landscape of cellular states. So many initial analysis approaches in single-cell RNA-seq data, definitely, a couple of years back, included clustering the cells into discrete clusters and performing differential expression on the clusters. Here on the left, I show you an example of a data set of neurons that were sampled from the retina, which was a work by Karthik, a former postdoc in Aviv's lab, where you can see visually discrete clusters, and such an approach seems reasonable to do. With our data, the majority of cells didn't form discrete clusters, but rather looked like a blob with some structure in it. And that required different computational tools to analyze this type of data. To identify transcriptional programs that describe the diversity in the data without having to cluster the cells, I turned to topic modeling, which was a method originally developed in natural language processing, which a former postdoc in the lab, Samantha Riesenfeld, spearheaded in the lab. So in topic modeling, documents are naturally described as mixtures of topics, which you can think of as themes a document talks about. And there is reasonable analogy between text analysis and transcriptomic analysis, where a cell is analogous to a topic and a gene transcript appearing in the cell is analogous to a word appearing in a document. A topic then in text analysis is represented as a probability distribution over words, shown schematically on the left. Note that we don't tell the model anything about topics ahead of time. But what has been found in text mining is that these inferred topics do have the property that they are often interpretable, that is, humans can associate a theme or semantic concept with highly weighted words in each topic. So going back to our analogy between text and cells, words appear in a document probabilistically from the topic distributions, just like the gene transcripts that appear in a cell results from the processes that are active in a cell. So what we get after running topic modeling is a distribution over genes for each topic, as well as a mixture of weights over all topics for each cell. We can think of the topics as biological processes, and it is up to the user to associate these biological processes with the topics, and where we heavily rely, of course, on previous biological knowledge. I'll show you now some of the results topic modeling gave us on our data and how it was useful for downstream analysis. Here you see the same UMAP embedding as you saw before for all anti-tumor CD8 T cells across all time points, and they're colored respectively by the topic weights. Furthermore, we can look at the distribution of topic weights across time points, as you can see here shown as an empirical cumulative distribution, and ask if any of these topics vary in their activity over time, and we can test this, of course, statistically, taking mouse replicates into account to test for robustness. Furthermore, we need to annotate these topics with biological meaning, and if the transcriptional diversity, we detect some context. So this can be done by looking at the top specifically weighted genes, as you can see to the left with the bar plots. Alternatively, we correlated the cell weights for the topics to signature scores using programs from the literature, which you can see here in this heat map to the right. And I'll come back to this heat map in a moment. We can also use the distribution over topics for analyzing the link between the T cell receptor and the transcriptional diversity of T cells. So our data was generated using the 5'10x technology, which gives us both the T cell receptor on top of the transcriptional profile for most cells. And we might be interested, for example, in asking how cells that share the same TCR, that is called a clonotype, compare to each other transcriptionally. To answer this question, I looked at all the cells of the clonotype and averaged their distribution over all topics, which is visualized in this heat map. On top, I indicate the size of each clonotype and whether a clonotype on average is uniformly distributed over all topics, or if it's more heavily weighted in specific topics, can be quantified with a Gini index shown in the top. And the higher the Gini index, the more non-uniformly the distribution. So here I show you two clonotypes as examples that populate very different areas on the UMAP. You can see in the heat map that particularly topic 12 seems to show non-uniform expression. As you can see, quite a few clonotypes that are pretty red for topic 12. And topic 12 was a TH17-like transcriptional program. So this analysis suggests that TCR might play an important role in CD8 T cell differentiation into a TH17-like cellular state. Going back to the heat map from before, we identified two topics, topic 10 and 12, which did not correlate or even negatively correlated with reference signatures of dysfunction and positively correlated with signatures of TCF1 positive, PD1 positive CD8 T cells. So by the way, the gene TCF7 is encoding for the protein TCF1. Something to keep in mind so you're not confused. We were very excited to find these cells in our model because these tumor-resident PD1 positive, TCF1 positive cells have been called stem-like or precursor-exhausted T cells, as you can see here in these figures of working models from many papers that all came out in recent years. According to the prevalent view, these cells are relatively undifferentiated, tumor-reactive CD8 T cells that infiltrate the tumor microenvironment and have the ability to persist and locally and give rise to fully exhausted TIM3 positive T cells. Furthermore, these cells have been suggested to form the cellular reservoir of tumor-specific CD8 T cells that are able to expand and give rise to an enlarged pool of differentiated cells in response to immunotherapy. However, as you can see in this graph to the right, the sheer fraction of TCF positive cells within PD1 CD8 T cells was no predictor for response to checkpoint blockade in human advanced melanoma. This suggests that the thorough understanding of the generation, the maintenance, and the differentiation of PD1, TCF1 tumor-infiltrating lymphocytes will be essential to predict treatment response and to fully exploit the therapeutic potential of these cells. Our topic analysis predicted that these cells would increase with time, and we validated this by flow, that indeed the percentage of TCF1 positive tumor-specific CD8 T cells increased. These cells peaked at 5 weeks in their ability to secrete cytokine and proliferate, but they don't upregulate inhibitory receptor expression after 5 weeks. So against the model of them being stem-like, we show that they are not resistant to tumor-induced dysfunction and are functionally very distinct between early and late tumors. The erosion of residual functionality of TCF1 positive tumor-specific CD8 T cells can arise due to either a homogeneous shift toward the dysfunctional cell-intrinsic program, or a change in the proportion of different cell subsets within TCF1 cells with different functional capabilities, or both. So to test this, we analyzed the subset of cells in single-cell RNA-seq dataset that expressed TCF7. We then applied topic modeling again on a subset of these cells which expressed TCF7, and we found that A, there was ample heterogeneity in these cells, something that was largely missed by previous studies on these cells, and B, that this diversity was dynamic over time, as you can see in the heat map, as red indicates an increase and blue a decrease in topic expression compared to 6 weeks. We then wanted to see whether the variation we observed also corresponded to profiles of CD8 tumor-infiltrating lymphocytes in human tumors, as estimated by a single-cell RNA-seq dataset of melanoma tumors. In several programs identified by topic modeling in our dataset correlated with topics we identified in the human dataset. So this approach represents an attempt to compare cellular states and transcriptional diversity across datasets, and in this case, even between species, which is a computationally hard task to do. So next, because previous TCF7 signatures failed to adequately recapitulate the heterogeneity within the TCF7 compartment in our dataset, we next interrogated whether this lack of concordance was due to variable expression of signature genes. So in this plot, you can see an approach at identifying feature genes, or often called variable genes. This is a method that was developed by former postdoc Adam Haber in Aviv's lab. So when plotting the fraction of cells that express a gene as a function of total count of a gene across all cells, it forms this nice sigmoidal curve. Genes that are outliers to the right of this fit are often of biological interest and can be used as features for downstream analysis, such as PCA. Interestingly, these feature genes, when we intersect them with a signature of TCF7 from the literature, we identified 34 genes marked here in red. And one of these genes was SLAMF6, a member of the CD2 immunoglobulin superfamily, which caught our interest. The single-cell data suggested the SLAMF6 was downregulated with time within TCF7 cells, which we validated with flow. Several topics were significantly differentially weighted as a function of SLAMF6 expression, suggesting functional differences between SLAMF6 positive and negative cells. And when doing differential expression between these populations, we saw that lower SLAMF6 expression was correlated with higher expression of genes encoding the chemokine CCL345, as well as galectin-3, an inhibitory ligand for LAG3, and the immunosuppressive cytokine FGL2. This suggested that SLAMF6 positive and negative TCF1 tumor-specific CD8 T cells may play different roles in modulating immune cells at the tumor site. To the left, we show that although most likely TCF1 cells are SLAMF6 positive, the proliferative capacity significantly declines with time. And to the right, you can see that SLAMF6 positive cells express higher levels of inhibitory receptors. So taken together, the SLAMF6 positive and SLAMF6 negative TCF1 tumor-specific CD8 T cell populations appear phenotypically and functionally distinct and may have different roles in promoting and maintaining the anti-tumor immune response. So where do these SLAMF6 negative cells come from? They appear around 7 weeks post-induction. So we were wondering, where they come from. Naturally, you might ask if they differentiate from the SLAMF6 positive cells and just lose the SLAMF6 expression. We didn't think this was the case because it's rare that cells downregulate inhibitory receptor expression while becoming more dysfunctional. So we looked at recruiting from the draining lymph node. To test whether these populations both home from the draining lymph node, we treated mice at 6 weeks post-induction, so just before SLAMF6 negative cells appear in the lung, with FTY 720 for 2 weeks, and this blocks egress from the draining lymph node. When we did that, we saw that specifically the SLAMF6 positive cells proportionally were affected by that, and not the SLAMF6 negative. And more specifically, the cycling SLAMF6 positive cells dropped out even more significantly. So this suggests that recently recruited SLAMF6 positive tumor-specific T cells may have greater proliferative capacity than SLAMF6 positive tumor-specific T cells already present within the tumor microenvironment. So in summary, I showed you how we could study TCF1 tumor-specific CD8 T cells in a chronic tumor model and saw their progression towards dysfunction and how their transcriptional diversity shifted over time. More specifically, we describe a shift from SLAM6 positive to SLAM6 negative cells over time, which are functionally distinct. And last, we showed that the tumor-specific CD8 T cells in the draining lymph node are a vital source of SLAM6 positive, TCF1 positive tumor-specific CD8 T cells for the anti-tumor immune response. In terms of my analytical approach, I showed you how we used LVA to describe the transcriptional diversity of the cells and how it allowed us to quite easily test how this diversity shifts over time, how it compares to other data sets, and study the link between the T cell receptor and gene expression. And with that, I'd like to thank again all my amazing collaborators and who made this work possible, and of course to Aviv for her support and encouragement throughout. And with that, I'd be happy to take any questions.
Okay, so we have a first question by Hanan. What are the product services Immune AI offers and to which type of customers?
So as I said, we're like, it's an end-to-end platform. So basically we receive blood samples and then we will generate all of the libraries ourselves and do the analysis of the data. We have collaborations with both academia and industry. So I think we're really open to collaborate with whoever.
Thank you. So far, no more questions. Anyone wants to ask questions, any question for Rebecca, now it's the right time. Another 10 seconds. Okay, so I guess no more questions. So thanks a lot, Rebecca.
Sure, thank you.
And now we'll move to our second speaker tonight, which is Omer. Omer Weisbrod is a postdoctoral fellow at Talcott Price Group at Harvard University. His research lies at the intersection of machine learning, statistics, and genetics. Previously, he was a postdoctoral fellow at Eran Segal's group at Weizmann Institute of Science. He completed his PhD at the Computer Science Department of the Technion under the supervision of Dan Geiger and the Statistics Department of Tel Aviv University under the supervision of Sharon Rosset. He was also a paradigm researcher at the Machine Learning for Healthcare and Life Science group at IBM Research Haifa, where he worked on a variety of projects involving analysis of genetic and clinical data. Today, he will talk about predicting genetic disease risk across diverse human populations. So, Omer, go ahead.
Open your microphone.
All right, can everybody hear me? And can everybody see the screen that I'm sharing? I will assume that the answer is yes. Awesome.
Yeah.
Cool, great. Thanks for having me here. It's a real honor. So yeah, I'm Omer Weisbrod. I'm a postdoc at the Harvard School of Public Health, and I will be speaking about predicting genetic disease risk across diverse human populations. So I'm going to start with a bit of a personal perspective. So this is a figure from the first paper that I've worked on in 2011. So we had a family of people and some of them had some genetic disease and some didn't, and we had their genotypes. And overall, we had 43 people. We thought it was a very impressive data set. and it was actually well powered, and we were able to identify the causal gene for that disease. And that was in 2011. And this is the data that I'm working on right now. It's called the UK Biobank, and it has around 500,000 people, which is kind of close to 1% of the population of the UK. And here you can see a map of the UK, and every black dot is the place of birth of one of the participants. So we had, or at least I personally had, an increase of 3 orders of magnitude in way less than a decade. And this is kind of representative of where genetics stands right now. So I just mentioned the UK Biobank, but this is only one of multiple biobanks that are being formed around the world. So we have biobanks in Finland, in Estonia, in Japan, China, Qatar, and in the US, they came up with like 3 competing initiatives because, you know, the US always manages to scrub their healthcare projects. And there are even discussions about a biobank in Israel, though it's always under discussion, as far as I know. And I will just briefly mention that all of this is just a drop in the bucket, because most of the genetic data that we have comes from these private companies like 23andMe and Ancestry.com and MyHeritage here in Israel, which have data of tens of millions of people. And as of 2019, it increased exponentially, though I'm hearing that it flattened since then. So anyway, hopefully I've convinced you that we have lots of genetic data. And then the question is, what can we do with all of this data? And I would argue that maybe the most immediate promise of genetic data is polygenic risk scores. So polygenic risk scores, the idea is very simple. We take a person's genotype and we use that to predict their phenotype. And this phenotype can be everything. It can be, you know, height or weight or cholesterol or some genetic disease. And this is something that people have been speaking about for decades. But over the last 2 years, this idea really started to materialize based on some really high impact papers. So the paper here at the top was very influential. So it showed that we can predict risk of heart disease just based on genotype. So we can do that for a person, you know, from birth. And the paper argued that people with extreme genetic risk should actually start taking statins way before they have symptoms, years in advance, and that could, you know, save their lives. So this is actually actionable. And this was in 2018, and in 2019, the same group basically did the same thing for weight and obesity. And you can see at the bottom that it also made it into the New York Times. And I happen to know that this week, a hospital in Boston started offering participants, you know, this service. So patient this service, so they can get genotyped, and then they can, you know, get a risk profile for heart disease. And if they're with extreme risk, they should start taking statins or be, you know, taken care of. And so, yeah, this polygenic risk score idea is starting to materialize. But these polygenic risk scores have a dark side. And the dark side is that they lose accuracy in non-European target populations when we use European training data. And this fact was made prominent by this paper you can see at the bottom by Alicia Martin et al. In 2019. So I'm going to show you 2 figures from that paper. So here on the left, you can see that most of the genetic data that we have in the world right now comes from Europeans. So we mostly have European genetic data rather than worldwide human genetic data. So that's a problem. And here on the right, you can see that when we train phenotype prediction models using European training data, and then we predict in non-Europeans, we get this huge drop in accuracy. You can see here that as we go from left to right, we get a larger and larger drop in accuracy, and it gets worse when we get to Africans. So you can see that for Africans, we usually get less than 50% of the accuracy that we get in Europeans. And that's because Africans are the most genetically divergent from Europeans among all of these ancestries here. And this is, of course, a huge problem for 2 reasons. So the first reason is a healthcare concern, because this means that we cannot predict risk for Africans as well as we can for Europeans, and this could, you know, exacerbate health disparities. So of course, it's a huge clinical and healthcare and societal issue that we should resolve. But the second reason is scientific, because in science, you want your models to generalize and to replicate across different populations and cohorts and settings, right? And you can see here that our models don't really replicate that well. and that means that we're not doing a very good job. So I think about these off ancestry predictions as kind of a stress test of what we do in the field of statistical genetics, because it really tests our models and our assumptions and our hypothesis. And this work is partially an attempt to investigate this, this loss of accuracy and to suggest solutions. All right, so I use the word polygenic a lot, so let's break it down a little bit. So the word polygenic means that a trait is affected by many variants. And to demonstrate what this means, I'm going to show you some figures posted on Twitter by a bot called GeoSbot. So you can follow it on Twitter, and every day this bot posts the results of one genetic study. So one genetic study per day posted on Twitter, and this is what it looks like. So I'm going to show you a few examples. So this is blood pressure medication. So what you can see here is on the x-axis, you can see different letters in the DNA, and the y-axis is minus log 10 p-value of the association with blood pressure medication. So the higher up a point is, this means that this letter is more strongly associated with blood pressure medication, meaning that people with one letter are more likely to take blood pressure medication than people with the other letter in this position in the genome. All right, and everything above the red horizontal line is genome-wide significant. So after correcting for multiple testing correction for the entire genome, so you can see that there is lots and lots and lots of places in the genome that are associated with blood pressure medication. So this is what we mean when we say a trait is polygenic. We mean that the signal is scattered all across the genome or all over the place. And it turns out that this is kind of the common pattern. So basically, almost every trait is polygenic. And I'm going to show you a few more examples, which I personally found exotic. So let's start with miserableness. You can see that miserableness is extremely polygenic. Basically, every place in the genome is associated with the property of being miserable. And this data, by the way, comes from the UK Biobank. So this is the UK. Basically, everybody's miserable anyway, but you can see that some of them are more miserable than others, and it's genetic. And a few other more exotic examples, hot drink temperature is also genetic. There is a genetic component, and this genetic component is polygenic. So some of the British people like their tea hotter than others. And cereal type is also a genetic polygenic type, though to be fair, most of the signal here comes from chromosome 6, where the HLA, autoimmune system related system is located, and that means that probably most of this signal comes from celiac disease. But anyway, hopefully I convinced you at this point that basically every trait is polygenic except for a few very special traits. And this observation that we started seeing around 10 years ago led to this tension in statistical genetics between two paradigms, which I'm calling the classic and the polygenic paradigms. So the classic paradigm says that we should study each gene or each variant associated with some trait separately. And this sometimes makes sense. For example, in psoriasis, you can see here that almost all of the signal comes from this one place in the genome, which is again on chromosome 6. So maybe it makes sense to kind of dig into this one place in the genome. And the pros of the classic paradigm is that it lets us really focus on biological mechanism, because we can actually look at what genes are in that position, and we can really dig into the biology and try to understand what's going on and what pathways are involved and so on. But the cons is that we're kind of ignoring what we're calling the global disease architecture. So it's kind of missing the forest for the trees, because we're not really asking questions of the type like, what is the the common theme for all of these genes that are related to the trait, or how many genes are involved, or how much does genetics contribute versus the environment to that disease, and so on. And so on the other end of the spectrum, we have what we call the polygenic paradigms. And the polygenic paradigm says that we should study the global disease architecture rather than individual genes. And more often than not, we see genetic results like you can see here for schizophrenia, where you can see that the signal is really all over the place. So schizophrenia is extremely polygenic, and this observation led to the polygenic paradigm. And the pros and cons are kind of the opposite from before. So the pros is that we can understand disease architecture because we can ask all of these questions that I mentioned before, like what is the common theme of all the genes and how many genes are involved, and what's the role of genetics versus environment. And but on the negative side, we're kind of losing biological understanding, because when we look at this in a very global, holistic manner, it kind of becomes a black box. So we can ask lots of statistical questions, but we don't really understand the underlying biology. And what I've been working on over the last few years is an attempt to bridge between the classic and the polygenic paradigms. And this is kind of a pretty wide framework that allows us to do lots of things. So for example, it allows us to improve phenotype prediction, especially out of ancestry, which is what I'm going to focus on today because I thought that's what's going to interest you. But it lets us do other things as well. So we can identify causal genetic variants. I'm going maybe to speak about it a little bit. And it lets us also do what I'm calling to dissect genetic architecture, which means that we can not only infer the genetic architecture, but we can also kind of dissect it into different components and ask how many genes contribute to that component of the of the trait, and how many genes contribute to a different component, and so on. So the way I think about it is that it's kind of a lens that lets us zoom in and zoom out of the trait. So we can zoom in and look at a very specific gene, or maybe even one specific SNP, or we can zoom out and look at the entire polygenic architecture of a trait. And what I'm trying to do is to come up with a unified framework that lets us zoom in and look at this at multiple levels of resolution. All right, and this is just a teaser or a spoiler. So I'm going to show a new method for phenotype prediction that we call polypred that adopts this unified framework, and that will improve our ability to predict risk by, for example, over 30% over the best method right now in Africans. But this is just a teaser. So I'm going to dig into this figure later on. So, all right, so the rest of this talk is going to be consist of these 3 sections. So actually, for most of the time, I'm going to speak about the first one, risk prediction using causal SNPs. And then I will build on top of that in the next 2 sections, which will include prior causal probabilities and combine data from multiple ancestries. But it's kind of these sections build on top of each other. So let's start with risk prediction, and let's start by saying, what do genetic datasets look like? So we have a genetic dataset, all right? So we have like around 500,000 people in the UK Biobank, and for every person we have their genotypes, which are 2 strings of letters, and usually we have around 20 million variants or SNPs for each person. It really depends on the frequency threshold, but let's say it's around 20 million. And for every person we have some risk factors, which could be stuff like diet or lifestyle, you can see here. And we also have their phenotypes, which could be anything. It could be height or BMI or cholesterol level or genetic diseases, right? So we have all of this stuff. And yeah, and now we're trying to relate all of these things to each other. And I will jump now straight into the water and present a probabilistic model that we're calling polypred. So, all right, so let's look at the model. So we have this trait Y, so we have a vector of around 500,000 people of the trait values, and we're assuming that Y is given by this linear model, X times beta, where X is a matrix of SNPs, so it has around 20 million columns, and beta is a vector of SNP effects, so it has 20 million entries, plus epsilon, where epsilon is a vector that captures environment and noise and basically everything else. I'm making it very simple on purpose. It's kind of more complicated in practice, but this is a good way to think about this. And now we're going to introduce an assumption that will make our life complicated. We're going to assume that each beta I can be either 0, meaning it's a null SNP, or it can be normally distributed, meaning it's a causal SNP. All right, so we already did a lot of things here. So we're already assuming that only part of the SNPs are associated or are causal for some trait, right? And we also just became Bayesians, and we introduced a Bayesian generative distribution, right? And this Bayesian framework lets us do lots of stuff. And I will speak mostly about 2 things. The first is what we're calling fine mapping, which is the task of identifying the non-zero beta i's, the relevant SNPs. So in this example, maybe only these 3 SNPs with the circle around them are actually causal, right? And so that's one thing. And the second thing is risk prediction. So risk prediction is we get the genotypes of some new individual, and then we're estimating this quantity, the sum of xi, beta I hat, where xi are the genotypes and beta I hat are the estimates of beta I. And the common task is that we want to estimate the posterior distribution of beta, right? So that's it. So we have this linear model and we have this Bayesian assumption, and now we want to estimate the posterior distribution of beta. So conceptually it's simple, but I'm going to try to convince you that it's not that simple. And this Bayesian model is going to do a lot of heavy lifting. So before moving on, I know that I gave these talks to similar audiences before, and I know that everybody's asking this question. So let's address this head on. So why do we use linear models? Because it's 2020. So I'm going to give you 3 reasons or excuses, maybe, in increasing order of relevance. So first is because genetic data sets are small. And what I mean by that, so I told you that we have around 500,000 people, but we have millions of features, millions of SNPs. And the important point is that we actually have millions of effective features, meaning that this data is not redundant. So the genome is not redundant. So we can't really compress it, or we can't apply an autoencoder that only extracts a small number of relevant features. We actually need millions and millions and millions of features to represent the entire genome. Maybe not 20 million, but it doesn't compress by an order of magnitude. And so we have many, many more features than samples. And that means that we can't really apply a brute force approach. If we just use a fully connected network, it's not going to work. We need to be smarter than that. The second reason is because inference is so much easier. So in genetics, we often want not just to predict, but also to do inference for all kinds of stuff, like which betas are causal, or how much does genetics contribute versus environment. And linear models make our life so much easier for inference, because for inference, we often want to compute confidence intervals and stuff like that. But the real reasons is that all other approaches that people tried don't deliver yet. And I know that because I'm one of these people who tried. So some brave souls have tried, and nobody heard back from them again. So it's kind of an open question. Can we dramatically improve our prediction capabilities using nonlinear models? Maybe, but so far nobody succeeded. Many people tried, nobody succeeded. And I've been thinking about this a lot. And if I have time in the end, if people are interested, I could speak about this a bit more. But for now, we're going to keep using this linear model, which beats basically everything else. And so going back to polypred, so I just said that we're trying to estimate the posterior of beta. So beta is our vector of SNP effects. So again, we're treating the SNP effects as random variables, and they have a generative distribution. And I'm going to now differentiate what we're doing, what polypred is doing, from existing methods for polygenic risk-scores. So what basically every existing method does is to choose a subset of SNPs, so a subset of columns in this matrix X, and then to estimate the betas using only the corresponding entries. And what this means is that each beta represents a combination of two things, a causal effect and what we're calling a tagging effect. And by tagging, we mean that each SNP aggregates the effects of all of the SNPs that were not selected, but are correlated to that SNP because of what we call LD. So we have these correlation factors between SNPs in the genome. And the problem is that these correlation factors are very specific to a population. So usually we do that using Europeans. So we get something that is very specific to Europeans, and that's why they don't transfer well to other ancestries. And so the idea of polypred is very simple. We're just going to do this for all SNPs instead of just a subset of SNPs. And then each beta actually represents a causal effect. So we're actually estimating what we would like to estimate. And that means that it should transfer better across ancestries. All right, so conceptually, I think it's extremely simple, but this is very challenging technically. And I can say that this is by far the most technically challenging project that I've worked on by far in my life. And we have computational challenges and we have statistical challenges because we want to do this on a very large scale. And I'm just going to give you kind of a brief taste of the type of challenges. So this is going to get a bit technical, but it's only for 2 slides, so bear with me. So let's speak about the computational challenge. So we want to compute the posterior distribution of these betas, but we cannot do it directly because it's computationally intractable. And to see why, let's think about the likelihood. So if you're used to think about loss function, so our loss function is the negative log likelihood, so the likelihood. So let's say that we already know which SNPs are causal. We already know that there is a specific set of SNP that is relevant. So the likelihood of the phenotypes is a multivariate normal with some mean vector and covariance matrix that looks like a sum of 2 things. So the first is what we're calling the genetic covariance matrix, which is proportional to XC transpose, where XC is the matrix X with only the relevant columns. And we have this noise matrix, and the bottom line is that if we want to compute the likelihood, we need to invert this huge matrix. And this is a 500,000 by 500,000 matrix that's intractable. There's no way we could do that, right? And usually if this were any other domain except for genetics, that would be the end of the story. But genetics gives us kind of a wild card that, or a get out of jail free card, which is recombination. So let's speak about recombination for a second. Recombination means that DNA is inherited in chunks. So here is an example. Let's say that we have this family of mom, dad, and child. So the child inherits one chromosome for the mom, and you can see that it's composed of chunks. So we have the blue chunk, and then the orange chunk, and then the blue chunk again. And the same for the dad. There is the purple chunk, and the green chunk, and the purple chunk, and so on. So this is recombination, and recombination has two consequences. So the negative consequence is that there is a very strong correlation between nearby variants, because most people who got something from the mom, the variant next to it, they also got from the same chromosome from the mom. And that means that in the population, variants that are nearby tend to be correlated. And this makes it difficult to identify the causal variant, right? Because if you have two correlated variables, it's difficult to tease them apart. But on the positive side, there is no correlation between distant variants, because this process decays very quickly. The correlation decays very quickly. So if you write down the correlation matrix between the SNPs, we get this banded matrix, meaning that when we get far away from the diagonal, we get zeros. And this is cool. So let me show you why very quickly. So we're going to impose a computational trick. So again, we need to invert this matrix, right? It's a 500,000 by 500,000 matrix. That's intractable. But now we're going to do something that is kind of similar to the kernel trick in machine learning. So those of you remembering their machine learning 101 days, remember that when we have to work with a matrix that looks like X transpose, we can instead work with X transpose X. And I just convinced you that X transpose X is a banded matrix. And now we're going to do a second trick, and we're going to approximate this banded matrix as a block diagonal matrix. And inverting a block diagonal matrix is easy. We just invert each block in turn. And if you haven't been following, let's just get to the bottom line, which is that we can decompose a problem into many easier subproblems because of this miraculous property of genetics. And this is just an example for the type of challenges that we have to address. We still have to compute these banded matrices. This is still a challenging problem, but it's manageable. And let's speak about the statistical challenge for a second, just to give you a taste. So we need to estimate the posterior distribution of beta, and it's hard. In fact, it's considered a super hard problem in statistics in general, when you need to compute the posterior distribution of effect sizes from this complex prior distribution. And we're using a method called SUSY, which came out, it was officially published actually only this year. It was published on arXiv around a year and a half ago, and I find it really beautiful. So I just give you a brief taste. So we have a generative model. So let's say that we have a true vector of effects beta. You can see it here, and the generative model says that beta is given by the sum of these one hot vectors. So you can see that we have a bunch of beta L's, and each one has only one non-zero entry, and the identity of which entry is non-zero is selected probabilistically. And that's good because we can incorporate biological knowledge into this. I'll speak about it later on, but it basically means that if one SNP is inside a gene and the other SNP is not inside a gene, then we can that means that we can we can give larger weight to the SNP that is inside the gene. So that's really cool. And that's our model. And our goal is to estimate the posterior distribution of beta. And very briefly, what SUSY does is it iteratively updates the posterior distribution of each beta L, of each of these components, conditional on the others until convergence, until we converge. And why does this work? It works because it turns out that this actually optimizes the variational approximation of the joint posterior of these betas as a factorized distribution. And if you're not following, it's not really important. I think that it's really helpful to look at an example and just to give you a taste of why this is beautiful. So let's say that we have our true vector beta. We don't know that that's true beta, right? We only have the data, the x's and the y's. And now we're assuming that we have these three components, and we're kind of going to iteratively refine our posterior distributions of each of these components. So this is what it's going to look like after one iteration, and this is after the second iteration. And after the third iteration, it's going to look like that. So you can see that now we have a fairly accurate approximation of each component. So basically, each of these latent vectors the posterior now represents one of the true effects in the true vector beta. So we can't know for certainty which SNPs are causal, but we know that probably one of the three on the left is causal, and probably one of the three in the middle, probably one of the, I don't know, the 3 or 4 on the right. So this is the idea behind this method called Suzy. I find it really beautiful, just to give you a taste of what we're doing. And now I'm going to kind of skip ahead, and I did all of this complex task. I estimated the posterior distribution of beta for each and every SNP in the genome, or at least the 20 million, most common ones. And by doing that, we have a prediction model, right? Because now we can take our beta estimates and to plug them into a prediction, one of the linear prediction models. And so we did that, and I'm calling this method polypred minus because there's still a missing ingredient, which I will tell you about later on. And so let me walk you through what you're seeing here. So these are the results. So what we did is we took 7 genetically uncorrelated traits that we had across multiple cohorts, and we trained a bunch of methods using only British people in the UK Biobank, so 337K British people. And then we applied the predictions on non-British people, including non-British Europeans. So you can see here the ancestries here at the bottom and the sample sizes. So we have non-British Europeans. This is kind of our gold standard. And we evaluated 3 methods. So we have our own method, polypred minus, and we have a method called BOLT, which according to our experiments is the best method that is out there right now. And P plus T is basically what most people are still using, but you can see that it's really not good. So I'm just including it here kind of, to demonstrate that. And you can see that polypred minus, this weakened version of our method, is not much better than BOLT in most ancestries, but it's getting better as we go to the right, and it's getting much better for Africans. And the reason is that it really exploits the cases where the correlation factors between SNPs is different from British to the target population, and it's most prominent in Africans. And yeah, I should also mention that the y-axis is showing what we're calling the European normalized relative R-square, and that basically means that we took the the R-square of the prediction accuracy and we divided it by the R-square that BOLT gets in Europeans. So you can see that BOLT in Europeans, by definition, it gets a relative R-square of 1. That's the left bar, the orange bar at the left, right? And so we normalized everything according to how well BOLT works in Europeans, and that's how we get our accuracies. And so you can see that we got a 25% over BOLT in Africans. That's the blue versus the orange bars for Africans. But you can also see that in absolute terms, the prediction accuracy for Africans is still really low. So, I mean, the only takeaway for me is that this is a very, very difficult problem, and we made some progress, but we really need to do better than that or to have more data than that. And maybe this is a good time to ask questions. These are, by the way, the traits that we analyzed. So you can see that it's a combination of lots of different types of traits. And we mostly chose them because they're uncorrelated, and we had them across multiple cohorts. And this is maybe a good time for questions if anybody has any. All right. So yeah, this was the meat of the talk, and now I'm going to show a few improvements over this basic model. So the first is incorporating prior causal probabilities. So the idea here is that some SNPs are a priori much more likely to be causal than others. And to see that, let's look at this example. So I have this stretch of DNA, and now we have 3 annotations. So annotation is a property of a SNP. So one annotation, for example, is coding, whether a SNP is in the coding gene or not, and intron, and conserved, whether a SNP is evolutionary in an evolutionary conserved region of the genome. All right, so we have these 3 annotations, and without knowing anything else, we can already say that these 3 SNPs are probably the most relevant SNPs for any trait because they're coding and they're in an evolutionary conserved region and they're not in an intron, right? So we can already say that these are probably the most relevant SNPs a priori, right? So these are, you can think about these things as features over the features because the SNPs are the features in our machine learning problem, and each feature now has a vector of features, in 3 features in this example, but hundreds of features in what I did in practice. So here is an illustration of how this works. So let's say that we have, this these results from a genetic study, and we know that one of these 3 SNPs here in the middle is causal, but we don't know which one. But now let's say that we know that this SNP here in the middle is coding and conserved and the other 2 aren't. So conceptually, what this means is that we have much stronger evidence for this SNP in the middle than for the other ones, because coding SNPs are more likely to be causal than non-coding SNPs, right? So this is the idea, and I'm not going to go into too many details. So what we did is we took around 200 functional annotations. So again, that means that for each of the 20 million SNPs, we have around 200 annotations. So 200 features per feature. All right, it's kind of, you can think about it as kind of a hierarchical model. And we use that to specify prior causal probabilities for our Bayesian model. So very briefly, we have the prior causal probability. You can see that on the left. And if you write down the math, it turns out to be proportional to the conditional variance of each SNP, conditional on its annotations. And now we're assuming this linear model on the right, meaning that the conditional variance is given by a linear combination over annotations multiplied by these parameters called tau. So this is just an assumption of how the variance can be generated. And then we need to estimate these taus for hundreds of annotations, because again, we used many different annotations. I didn't mention what these are, but these are many different things. Lots of annotations coming from all kinds of high throughput biological experiments. So for example, stuff like whether a SNP is chemically accessible, because of open chromatin and histone marks and all kinds of methylation properties and stuff related to whether a SNP is in all kinds of evolutionary conserved regions in other mammals and so on. So this is cool because it's kind of an arbitrary, flexible framework. Whatever data that you have about SNPs, you can incorporate into a framework. It doesn't make any specific assumptions, and you can use that to improve the prior causal probabilities. And anyway, we did that, and we can estimate these taus using the method of moments. I'm not going to get into the technical details. I will just say that this is actually equivalent to multiple kernel learning from kernel machines in machine learning, but with hundreds of kernels, which is something that I haven't seen done in machine learning. So in that sense, I think it's kind of an open problem considered in machine learning, and this is a solution that is specific to genetics because it exploits properties that are specific to genetics. These are linear kernels because for all kinds of reasons, I don't think it makes a big difference. So yeah, anyway, we have these taus, and I'm going to jump straight to the results again. And when we do that, we now have the method called polypred. So polypred is kind of like polypred minus, but now we use these prior causal probabilities. So you can see the figure is similar to what I've shown you before, but now we have this new method polypred, the blue bars, and you can see that the blue bars are now always higher than the others. And in relative terms, this is a big improvement. So for example, in Afrikaans, we now get a 32% improvement over BALS, over the best method that is out there right now. But again, in absolute terms, the prediction accuracy for Afrikaans is still really low. Yeah, so I mean, this just goes to show what a difficult problem this is. And again, this is a problem, a healthcare problem and a scientific problem. And we also applied polypred on other cohorts to make sure that it generalizes. So we applied it on a cohort in Japan and then a cohort in Uganda. And we got similar relative improvements to what we saw in the UK Biobank. But in absolute terms, again, the accuracy is not that high. All right. So I will just briefly mention that this framework can be used not only for prediction, but also for doing other stuff. Because when you can estimate the posterior of each and every beta, the effect of each of every SNP in the genome for some trait, you can ask many different questions. So for example, we also did that to identify causal SNPs with a large posterior distribution of having a non-zero effect, a non-zero beta I, and we found lots of SNPs. That's what you can see here. And this lets us, as I've said in the beginning, to zoom in on specific parts of the genome and try to understand the underlying biology. So for example, what you can see here is that we found lots of places in the genome with causal SNPs, over 3,000 across many different types of traits because we applied it on many different types of traits in the UK Biobank. But one, for example, cool thing that we found using this framework is that many SNPs are causal for multiple genetically uncorrelated traits. So here is one example. There is one SNP in the genome that is causal for height, for BMI, for waist-hip ratio after adjusting for BMI, for red blood cell count, for total cholesterol, blood pressure, lung capacity, and balding. These are very different traits. They have completely different biology. They have nothing to do with each other, or so we think. But yet this SNP is involved with all of them. So that's a cool finding, I think. We don't really understand why, but I still find it a cool finding. All right, so the last thing is really short and simple, actually, combining data from multiple ancestries. So I kind of showed you what is the best that we can do right now, how well can we predict using only British people. So the way to improve that is to extend our method to use training data from multiple ancestries. So this is what we did here. So we can take a large European sample and a smaller non-European sample and to combine both of them in the same PRS model. And the idea behind our extended model, probabilistic model, is that each SNP now, instead of having a single effect, a single beta I, it's going to have multiple beta I's, one for each ancestry. So it's going to have a European effect size and an African effect size and so on. But we're going to assume that there is some sharing going on. And the way it's going to work is that we're going to assume that we have for every SNP a vector that follows this generative distribution. So either it's normally distributed with some covariant matrix sigma with some probability, or it's zero otherwise. And this means that every SNP, these effects can either be all zero or they're either all non-zero. So this is kind of how we regularize or constrain this model. And the matrix sigma says that if two entities are similar, then the effects are supposed to be more similar to each other. So the African and the South Asian betas are supposed to be similar and so on. And again, let's skip over all of the technical details. So we did that, and then we took training data from two different cohorts. So we took training data from UK Biobank British people, you can see here, and from Japanese people from Biobank Japan, another biobank. So we had lots of training data, and then we combined these two things, and we had this new model called Polypred Plus, and we applied the predictions on East Asian people in the UK Biobank, and we got a nice improvement of 24% over the state of the art. So combining data from Europeans and non-Europeans is obviously a good idea, and this is a principled framework for how we can do that. And all right, so I'm going to wrap up. So we proposed 3 models. So Polypred minus, which can estimate causal effects for the entire genome, and Polypred, which improves upon that by also using functional annotations, and Polypred plus, which improves upon that by also combining European and non-European training data. And I showed you that you get nice relative improvement, so 32% improvement over the best method that exists right now for predicting risk for Africans, and 24% for predicting risk in East Asians using Polypred plus. And I want to thank many wonderful people, especially my postdoc mentor, Alkes Price, and Wenbo Shi and Masakanai, who are working with me on this project. And thank you.
Thanks a lot, Omer. We have one question from Nadav Brandes.
Sure.
So you mentioned a single variant which is causal for 7 different traits. How can you be sure that this variant is really causal?
That's a good question. I mean, it's kind of a loaded question. So I mean, let's break it up. First, by saying causal, I mean that the posterior causal probability is larger than 95% for each of them. I don't remember the exact numbers, but this still leaves, a margin of error of maybe 5%, right? Assuming that the model is perfectly calibrated. So, by saying causal, we mean high probability of being causal rather than deterministically causal. That's one thing. Second thing is that the term causal itself in statistics and in statistical genetics is a loaded term because we're not really demonstrating causality in the very strict sense, right? For example, maybe, there is some effect of population factor that we haven't managed to account for. Maybe people in North UK have different genotypes, but they also eat different food. And so we kind of confound these two things with each other. I mean, we can't really, prove that in a very strict sense, but I meant that this SNP gets a posterior causal probability greater than 95% for all of these different traits.
We have a second question from All. Amazing talk. If I understand, you combine the British data with Japanese.
Yes. Is that the question? If that was the question, then the answer is yes. Yeah. Thanks.
Same person, and then you show the effect on East Asians. How does it perform on others?
Yeah, that's a great question. Not as well as we hoped. So, East Asians is mostly Chinese in the UK Biobank, and Chinese are genetically pretty similar to Japanese, and that's why we get this huge increase in Chinese, and we don't really get a nice improvement as we hoped in the other ancestries. And I mean, we're still looking into why, and this is, I should mention, a preliminary result. But yeah, it is disappointing that we kind of managed to improve our what I guess the tagging PRS, but not the ability to better fine map these SNPs as well as we hoped. I mean, I'm still hoping that we'll be able to fix that. And if there aren't any questions, I can just mention that I have this backup slide about the nonlinear models that I kept in case anyone is interested in that, but it's up to you. About how we can improve upon that using nonlinear models.
We have 2 more questions.
Sure.
From Nadav again. Do you have an intuition why nonlinear models aren't really doing so good in the field?
All right, so that's an excellent question because that's my next backup slide. So I'll answer that when I get to the backup slide. Anything else?
Yeah, one from Ranayal. Only SNVs or also indels and complex variants are included?
Yeah, that's a good question. We have SNPs and we have short indels. And the reason is that our data is based on genotyping, not on sequencing. And so, I mean, it's imputed data for those who are, who know what I'm speaking about. So we can only impute SNPs and short indels, not, copy number variants and stuff like that.
I think that's it for now. Although
All right,
said
so
there's a second part, but I don't see the second part.
Sorry, say again?
You can continue.
All right. Yeah, so somebody asked me this excellent question, why do you use linear models? So I prepared this slide especially. Yeah, so I mean, I kind of said this before. Show my screen. All right. Hopefully you can see my screen. So, the brute force approach is to kind of create a fully connected network. Everything is connected to everything. This is never going to work for, for our current genetic data sets because our genetic data sets are small. Even 500,000 people is small compared to the effective number of features. This is what I said before. It's kind of a brute force, barbaric approach. We need to be smarter than that. We need to, the way I think about it is that we need to remove some of the edges from this graph, right? Not to have everything connected with everything, because this is just going to overfit. So we need to find some modules, some way that makes sense to decide what interacts with what, not everything interacts with everything. And so I think that we need to incorporate lots of biological knowledge and understanding into these things. And I can speak about 2 ideas. One is that I'm very familiar with because this is a previous paper of mine that I worked very hard on, and it worked marginally well, but not as well as I hoped. So what I did here was I identified groups of SNPs that might be interacting, and the criterion was that they had to be physically close. They had to be nearby, so physically close on the genome. And then I kind of designed a nonlinear modular network for each group. So you can see, for example, these 2 blue circles at the top are 2 SNPs that are close by, and then I'm assuming that they interact. And then I made these non-linear networks interact additively. So I kind of linearly combined these non-linear networks, and that's my paper from 2016. It worked nicely. I don't think that's the way to really capture all of the non-linearity that's going on. That's kind of a heuristic. And what I'm thinking about right now, and it says work in progress, but I'm just thinking about it. I don't have time to actually do anything about it. I mean, I started, but I don't have time. I think that maybe the best way to do that is to actually incorporate real biological knowledge, not just SNPs that are close by to each other. So we have SNPs associated with different biological pathways, and I think that maybe the way forward is to kind of do the opposite of what I did in attempt 1 to construct an additive module of SNPs, for example, in every biological pathway. And we can do that using polypred, because polypred, for example, it has the beta for its SNP. So every pathway is just, it can construct a feature for every pathway as the sum of xi beta I for all the SNPs in that pathway. And then we need to have these modules interact non-additively, which I think makes much more sense, because this is what biology looks like. Biology is messy, and we have these pathways that interact with each other in all kinds of messy, non-linear ways. But the pathways itself, I think that we can model them as linear, not because they're truly linear, but because we have so many SNPs in each pathway that for the same reason that I mentioned before, because of the sample size, we could just as well model them as non-linear. So it's kind of a dimensionality reduction idea, if you want to think about it like that. I'm kind of reducing the model to a small number of dimensions based on biological pathways. And this is kind of an idea that I started playing around with, but then COVID happened and I didn't have the time. But I don't know if anybody's interested, I can speak about it offline or something.
So Lior is asking, why not use convolutional neural network, one-dimensional CNN?
Because I don't think that the properties that CNN are designed for work well for genetics. CNNs are designed, for capturing specific properties of images, right? You have translational invariance. You have all these properties that are specific to vision. I don't see any reason why they should work for genetics. I mean, maybe what they refer to is that there are lots of works in genomics that use CNNs and similar architectures. So this is kind of different. Genomics means that we want to predict some property of some SNP based on its neighborhood. I don't think that these assumptions of CNNs make sense for predicting phenotypes from genotypes. I mean, that's exactly what I mean. I mean, I think we need to be smart and to come up with the correct architecture, not just borrow architectures from other domains and just blindly hope that they work.
Thanks. We have two questions from Amir Livnet. First one, where do you cut the matrix to blocks? Do you use starts, ends of genes?
Sorry, seconds.
Where do you cut the matrix to blocks? Do you use starts, ends of genes?
Yeah, that's a good question. So I didn't give the full story. So the way I'm doing it is basically I'm not cutting it into into disjoint blocks. I'm actually using overlapping blocks. And so there is some overlap. And so for every SNP, I have multiple estimates of beta I's. And then what I'm doing right now, I'm just taking one of them from the block where it's closest to the middle of the block, to the center. But I could also do something fancier, take some combination. So these are these are overlapping blocks. And yeah, I just did them based on region sizes, so 3 megabase long regions.
Namir has a second question. Once you reduce a trait to depend on a small number of SNPs, could you add interaction terms between these few SNPs to your linear model?
I mean, my answer is that I don't really do that, but if I did that, then the answer would be yes. Because, I mean, I said that I'm finding cause of SNPs, but usually, I mean, I'm not sure what it meant, but we don't really find a small number of SNPs for each trait. We basically use millions of SNPs for each trait.
Any more questions? Okay, so it seems like we ran out of questions. So thanks a lot, Omer, for this fascinating talk.
Thank you.
So I'll conclude and say thank you to all the participants for joining this virtual event, and for of course Omer and Rebecca for these wonderful talks, and also Immune AI for helping to arrange this this professional virtual event, and lastly to Zoal from GetWebinar, helped us in arranging the event. Actually, there is one more question. All the data is open. I think it's for you, Anna.
Not really. So, genetic data, so there are privacy limitations. UK Biobank is relatively open in the sense that every researcher can apply to get access to the data, and they just they accept basically all applications. So if you're a researcher and you apply, you're probably going to get access to the data, but you can just download it right now. And this was actually a game changer. I mean, I don't have time to speak about it, but until the UK Biobank, genetic data was considered something that is really, really difficult to get. This really made a huge change in science, I think, by just having a huge data set, genetic data set that almost everybody can get access to. This made a huge, huge difference in genetic, science. I think this is a really big deal. I should mention that. I should give them credit for that.
Yep. Okay, so I think we're done. So thanks a lot.
Awesome. Thank
See you
you.
in the next event.
Yeah.
Bye-bye.
Thank you. It's been fun.
Thank you so much.